Can you put this down? Here, this one I want. Yes. Good afternoon. My name is Claudia Russo, and I am an associate professor at the Federal University of Rio de Janeiro in Brazil. I'm going to talk to you about building trees with MegaX and a little bit about alignment also. So I'm going to pick up from where Brian left us. And so let's build phylogenes in Mega. Well, the most commonly used method to build the phylogeny is press the enter key as many times as you need until a tree pops up on the screen. When it does, you just print out and then submit the paper. But there is an alternative. Well, define your problem very well. Select taxa and markers, and then you download the sequences. Perform a multiple alignment, which will be useful to establish homology among parts of different sequences. Tree building, so build the best possible tree. Topological tests to verify consistency of clades. And then your actual intellectual part of your paper start. So you start interpreting your tree and you start extracting biological information from your tree. So that's where it all begins. So how to do an alignment in Mega? So first of all, so we need to find out which part of one sequence is homologous to other parts of other sequences. And homology is not equal to similarity, although many people actually confuse these two concepts. Homology is similarity due to common ancestry. So two sequences or two parts of an organism are homologous of organisms, are homologous if their similar parts are there since their common ancestor. For sequences, we assume that the higher the similarity between sequences, the higher their chance of being homologous, such as this alignment down below. So an important note is that performing a multiple alignment does not test for homology, it assumes homology. So when you select your sequences, you need to be able to assume this homology before you run the multiple alignment. In this case here, this alignment was performed on purpose. So I selected, for these 6 birds, I selected different mitochondrial genes for each species on purpose and ran the alignment to check if my alignment program would crash or warn me. These are not homologous, but it doesn't. Actually, it does perform the alignment and the best alignment possible for this non-homologous sequence. And if you look at it in the distance and you run a tree with it, it will run the tree, and of course the tree is meaningless because there's no homology there. So there's no historical information in comparing the sequences. So it's important that you know that your sequences are homologous before you test the homology. So in this case, this is a bad alignment because this is the envelope gene for the HIV virus. So here I have a protein coding sequence, and here my alignment program put a gap here at this position for 7 of the sequence. And for the 8th sequence, it puts an alignment here. So that means that my alignment is perfect in this side and it's perfect on that side. But here we have a problem. So my one solution to this problem is always to use codon-based alignments. That means that you have your nucleotide sequences, you translated them into amino acids, you align these protein sequences, and you translate it back to the nucleotide sequences. So then you avoid those problems that I mentioned before. So here it would be something like how the program will do this. So in Mega, you would do this, well, start building your alignment, such as Brian told us to retrieve sequences for a file. And here, Mega gives you 4 options of alignment: align by cluster or muscle, regular alignment, or you can align by cluster using codon alignment, codon-based alignment, and you can align by muscle also using the codon-based alignment. So if you run this using some of the default parameters for the alignment, you'll have the final alignment here. This is for an example file for Mega. So about these parameters, well, we usually don't modify those, and people say that if you modify the alignment parameters and your alignment changes, is that because that illustrates that the poor alignment and the poor homology hypothesis for your sequences. Because if the homology is straightforward and that means that your sequences are very similar, then modifying these parameters for the alignment wouldn't modify the final alignment anyway. So we should just use the default. So, and now after the alignment, we should find out how these sequences evolve. So we have different models that may illustrate how well or how your sequences evolve. And one of the simplest models is the Poisson model, also known as Jux-Cantor or one parameter model. It actually, this is the actual divergence between two sequences. So this genetic distance starts from 0 and goes all the way to infinity here. And this is the observed difference, the percentage of the difference that you observe. See that it flatlines here at 75% by what Brian was telling us about, because we have 4 different nucleotides. So to independently produce sequences with 4 different nucleotides will average be similar at 25% of the residue, or dissimilar at 75% of the residue. So actually, this I'm going to talk about. So estimating this observable differences is actually useful. So to determine how well are into this flat line your sequences are, or how divergent these sequences are. This is the distance formula that's used for distance-based methods, but this model may be used for likelihood and other methods also. A more complex model would be Hasegawa et al. Actually, there are many. I just selected a few here. So this model is more complex. It has more parameters that are estimated from the data, such as the proportions of A, T, C, and G's, different rates for transitions and transversions, And it should be used if your sequences are more divergent than those that you would use for the one parameter model. And the favorite model for everybody is the GTR, which uses many parameters, the ATCG plus rates for all nucleotide transitions, and would be the highest divergence between the sequence that would be used. On top of those, you may use or not, you may or may not use the gamma parameter, which measures how similar are evolutionary rates when we compare different sides of your molecule or of your alignment. So the highest, well, this would be a curve, this would be a graph on the different rate parameters here. So if you have like just about one rate, all the rates would be regular in a sort of normal distribution here around some kind of mean. But if your sequences are, if rates among different sides of your sequences are very divergent, then you'd have like a shape such as this one, the green one. So the smaller the gamma parameter, the more you need to correct for those. So gamma is not another model, but it's something that you place on top of your model. So you can use GTR plus gamma, you can use a Poisson plus gamma, and etc. So selecting models in MegaX, you can do it manually. We will use the Explore Active Data. This is a very useful tool. And compute pairwise distance first in the distance menu. So I was telling you about that estimating p distance is very useful, because sometimes you'll see here, these are distances for Drosophila flies, and for the ADH gene. So here we can see that my sequences vary, the distance between sequences vary from 0.6 to maybe 0.21, 24 here, 25. So these are regular, are not so variant sequences. So that would put me in a curve, in my curve, in which the observed difference, which is this distance matrix, would be more or less what exactly happened if you consider multiple hits, multiple substitutions, backwards, and convergent substitutions. So I advise, even if you're not using a distance-based method, to estimate distance to avoid placing a sequence that is too divergent. For instance, one sequence that has a distance of 0.7 or 0.65, then you would spot it right here. But then going back at the Explorer, the Sequence Data Explorer will bring you to this window. And in this window, you can do a lot of things. You can unselect taxa, for instance, that you have noticed that the sequence is very divergent from the others. You can unselect taxa, and if you unselect it here, you will exclude it from all the subsequent analysis that you would make. You can edit and select taxa groups. For instance, here I have created a taxa group which I call Hawaiian Drosophila, and I place it Drosophila finidjunta, heteroneura, mímica, diastole, nigra, crassifemur, and albovitata into this group. And now Mega will see it, will see those taxa as as members of this Hawaiian Drosophila group, and I can use this to estimate distance between groups and distance within members of a single group and compare those averages, for instance. I can edit and select genes in domains. Here, I can add a domain. For instance, I could, if my sequences are a protein coding sequence with introns and exons, I can tell Mega that some portions of my sequences are non-coding and other portions are coding, and I can eliminate some of those when I'm gonna do the phylogeny, when I'm doing the phylogeny. I can, by pressing this button, I can see all my sequences without the identical symbol. I can translate it to amino acid, so I can check if the translation is perfect, as it should be, before I run my analysis. I can compute the number of conserved. Here are the number of conserved sites out of so I have 421 conserved sites out of 762. Number of variable sites, parsimony informative sites, and I have different types of degenerate sites. So 0-fold degenerate site, 2-fold degenerate sites, and 4-fold degenerate sites. Here's the 4-fold. And I can compute some statistic. For instance, nucleotide compositions that for a protein coding sequence, I'll have it species by species on the lines here, on the different lines. And I'll have overall and separately for all 3 codon positions. You should notice that the 3rd codon position are the ones that vary most, so you have to be more careful about this one. And you can use this to select a proper model, so that if these numbers vary a lot, you should select an evolution model that would take into account the different base compositions, nucleotide compositions. I can estimate nucleotide pair frequencies that will also be useful, selecting different transition transversion ratios. So here it's the same average for all positions together, and separately for the 1st, 2nd, and 3rd position. And here are the pairs. So this is when I have a T in one sequence, how many times I have another T in the other sequence, comparing pairwise data. And here, when I have a T, I have a G in the other, I have this number. When I have a C in one, a T in the other, and so on and so forth. And then I can use this data if transitions are more frequent than transversions. I can use this to select the model that would take this into account also. And the codon bias also. This statistic will give me the relative synonymous codon usage. So we would expect, if things were random, that if you had like 4 different synonymous codons, each one of those would be used around 25%. But this is often not how it happens. So this statistic will gives you a portrait, will illustrate how different from the expected average that you would have. And Mega does also for you, it does that automatically. So finding the best DNA and protein models in the EML, you do this by selecting this menu, just the model menu, and then you just You just press it and you have the result. With the top result would be the best choice. Not necessarily the highest parameter, but usually one of the top parameter used. So in this case, for this Drosophila, the Tamura Na93 plus gamma would be our model of choice. So the tree, you can construct, you can build the tree in Mega. You can open previously saved or a user tree in Mega, and you do analysis with it. Or you can use your data to generate, for instance, likelihood-based trees using in which you have a given tree and a model, statistic model, and you estimate the probability of observing the alignment based on these two things, the tree and the evolutionary model. So what the algorithm does is, if you have the model selected by previously, it will run by all the trees, all the possible trees, using heuristic algorithms and varying branch lengths, and to determine the tree that would fit your alignment best. You would do this using the phylogeny menu, construct and test maximum likelihood trees. So you may use Jukes-Cantor model, which was the one that I explained previously, the Kimura 2 parameter that corrects for transition and transversion ratios, Tamura 3 parameter, Hasegawa, Kissino, and Yano that I also talked about, Tamura Na, and the GTR model. So you can select each one of those here, and all these boxes are selectable items in Mega. So you can select the substitution model, you can select rates among sites, you can use the gamma distributed, you can add or not invariant sites to it, would be another category. You can, here you can include first, second, third codon position. You can sometimes, when third codon position are saturated, you might want to exclude third codon position, then rerun your tree to check if homoplasy is not interfering in your final tree, sometimes lowering bootstrap values. Use or not your known coding site that you had already selected as such in that data explorer. And you can select the heuristic method for the maximum likelihood tree. The maximum likelihood tree method algorithm is not exhaustive, so it's also always heuristic. So you may select a less exhaustive method, such as the NNI, nearest neighbor interchange, or more exhaustive and time-consuming methods, such as the SPR level 3 and the SPR level 5 would be the most exhaustive here. You can also select how you're gonna input the initial tree for the maximum likelihood. That means maximum likelihood algorithm will start from a tree and start rearranging branches on that tree and computing likelihood values and selecting those trees with highest values. And you can here select the type of the initial tree for the maximum likelihood. The default is the NJ and BioNJ, but you can select maximum parsimony, only neighbor join, only BioNJ, or you can input your own tree into that, or use the topology editor. So when you computing the ML tree, if you don't use the bootstrap, then this doesn't take that long to run this. But if you use the bootstrap, it might take a long time to run to compute the maximum likelihood tree. And this will be the tree, how Mega presents the tree. And here Mega automatically generates the tree caption with all the methods that that were used to generate it, and give you the reference as well for the methods. You can also use the distance-based methods, in which you take your alignment and transform it to a distance-based, a pairwise distance matrix, and use this to build the tree. For instance, the neighbor join algorithm, that you start with a star tree and isolate two neighbors and compute the total sum of branch lengths in this tree, with the 1 and 2 separated as neighbors. And you compute this S 1 and 2, and you calculate all S's for all possible neighbors. That means I'll compare the S1 and 2 with S1 and 3, S1 and 4, and so on, until all possible pairs are compared. And I'll select the pair of neighbors that minimize S. And then I'll group that pair into a single compound OTU and repeat all the steps until the tree is fully resolved. So here in the first step, I have the 1 and 2 joined. The second step, I have the 5 and 6 joined. And then the third step, I have the 1 and 2 joined with the 3, and so forth until all the tree is completely resolved. So Mega can estimate the neighbor join tree also. You can perform a test of phylogeny, and I'll talk about this more on the day after tomorrow, on Wednesday. And we can use the bootstrap or the interior branch test. We can also select different substitution models or the p-distance. We can include only transitions, only transversions, or both kinds of substitutions. We can also include gamma parameter. We can select complete deletion, pairwise deletion, and partial deletions are different ways of treating gaps and missing data on the alignment. Can also select one first, second, and third codon position and non-coding sites. And voila, the neighbor join tree. So here's the tree caption and the references. And you can press this button here to root the tree. In this case, Scaptodrosophila lebanonensis would be the outgroup. So if you press this when this branch was selected, it will automatically root the tree on this branch. You can also start looking a little bit more into your tree and presenting it to the user in a way that it passes on more information than only the topology and the names. For instance, here, if I select this interior branch and press this button, this window will pop on the screen, and I can place the name Hawaiian Drosophila here, and this will automatically give me a tree with this Hawaiian Drosophila shown here. That would be useful for my discussion in my paper, etc. So, and Mega has many different kinds of tools to present your tree. So you can input images, you can color code different branches, and I'll talk about on Wednesday. I'll talk a little bit more about this. So, thank you. Any questions?
Actually, I asked previously, but so one question is you have the new. Usually we have complete deletion and pairwise deletion. Now the new version has a partial deletion. I'm not sure the difference.
Yeah. So do you want to talk about this?
Sure. I mean, the partial deletion is basically when you are doing any analysis, you may contain sites which have lots of missing data. And so you may say, well, I will only want to use sites that have at least 50% of the sequences have a valid base. So then you can say, my cutoff is 50%. So that means Mega will actually exclude all positions that contains at least 50% missing data.
That's partial deletion.
That is partial deletion. If you say, my cutoff is 95%, then it will remove all the sites that have 95% or less coverage.
That'll be close to complete deletion in that case.
Yeah, and complete deletion is basically removing any position that has any missing data. So that's like 100%. So every sequence in that site has to have valid base. So as you generate these big phylogenetic data sets, which contains huge number of missing data in different parts of the alignment, it's good to maybe select 70%. So 50-50 minimum, but I would say 70%. So at least 70% sequences have a base at each position. You will be surprised that most of the data sets that are published in big journals, if you impose a 50% cutoff, have not many sites, maybe 1,000 sites. And they look really big, but they don't really have any information to resolve big things. So it is very, very good to check your result at different cutoff level. So this way, missing data did not have a big impact on the input.
How to set the values?
So if you select partial deletion,
Yeah.
you will get a new option, where you can say 75, 80, 30. It's up to you, what you feel is comfortable.
It's also useful because when you have gaps, sometimes you have more variability there and the alignment is not that good. So if you remove these sites, your overall alignment may improve.
Yeah, I think the 50% cutoff is a good cutoff minimum. So you can remove most of the positions that are missing in most of the sequences. There's no point having data that is so wholly missing.
There is another option for alignment other than Mega, and that is a program called Guidance. It's a web-based program. When you ask yourself, I've got this alignment. How good is the alignment? There's no internal way to answer that. And what Guidance does is take your alignment, put it in Guidance, and it evaluates it, goes through and it colors various regions according to the quality of the alignment of that region. It also colors sequences. You may go through and it says, you just don't want the sequence in here. It just misses matching anything so often. Say, okay, if I take that sequence out, my quality of my alignment improves. You may say, you know, there's this part of it, it's terrible, it just doesn't align very well. Depending upon what your purposes are, you may be able to just cut that out. And guidance gives you nice tools to be able to say, take this alignment and cut out this badly aligning region. So it's not a substitute for, but it's an additional tool that you can use, and just Google guidance alignments to get there, yeah, GUIDANCE, and use that to get some idea of the quality of the alignment and where the good parts. Often, you don't get any more robust tree, and it may be worse by including regions that align badly. Now, if what you're doing requires that at the end you have the complete sequences, you really can't do that, but it depends upon the purpose for which you are making the alignment and making the tree.
Yeah, I think there are many different ways of doing it well, and if the tree is your final destination, then you really need to do many things. If the approximate tree is okay for you, it just gives you some information about the data, then in that case you may not actually spend so much time with many things. Do a guidance or a mega at different cutoff levels, you should be fine. But if you're trying to establish phylogeny of a set of species, and there are controversial parts, then it's very much more complex, the whole thing. But anyway, yeah, to guidance, there are a few other software packages, and there used to be a G-blocks in the past and some others that you can use to evaluate how good the alignment is. Do you have a question? Cool. Anyway, if you have any questions or you want to see how this works, we have the computer here, and Christina or Claudia can show you how this stuff works. Okay, cool, thank you.
I know you already told us, but I think I missed it. Select competition. I didn't understand what it was. Select competition. That's what I said. I didn't understand what that was.
Yeah, because I was on the protein.